Hello, folks. We are happy that you're here. We'd like to welcome all of our members, friends, visitors. Uh, we have what I would consider a very interesting speaker, Dr. Kelsey Bentley. And rather than take up any more of this valuable time, I'd like to turn it over to Kelsey and let her explain exactly what she's been doing. Kelsey, would you do that, please?
Yes, hi. Thank you, David. I appreciate you guys having me out, especially because I think with the Australian whites having some hair influence, I hope that a lot of us will glean something from this presentation, being that majority of the work was done in Katahdin sheep. You will see that, I kind of call this the milking more from the post-weaning fecal egg count estimated breeding value. So this is kind of a combination of a national sheep improvement talk along with a colostrum talk. There's some response to vaccination, and then finally a bacterial challenge as well. So we kind of jump all over the board, but there is a singular thread that kind of traces this story all together. But first, maybe for those of you who don't know me, just a real brief introduction. Originally went to North Carolina State University there in Raleigh. I'm originally a micro native, though. No one knows where micro is, so I always love to put it on the map. In 2021, I met Dr. Scott Baldridge and went up to West Virginia University there in Morgantown to begin work on my master's. With my master's program, I really fell in love with the research and had more questions. I applied for the Ruby Doctoral Fellowship and received some funding to stay on. So that's how I ended up getting my PhD. And now finally, I'm landed in my current role here at Kansas State University as an assistant professor, where I actually have a three-way appointment. So I'm 60% extension, 25 research, and 15% teaching. So I'm a little all over the place, but I wouldn't have it any other way. Getting into really my research background, just to kind of set the stage for those of you who maybe don't know what some of those fancy terms I had in my title mean. An estimated breeding value is a science-based, industry-tested measurement of a heritable trait. So for this particular talk, I'm going to refer to post-weaning fecal egg count, but an estimated breeding value can be something as simple as weaning weight or yearling weight or maternal milk. So, all things that we can have a genetic correlation to. So, the PCV or PFCC value is actually a little bit on its head. So you can see here, I love to give the example of it's reported in percent reduction. So negative values are going to be favorable. So you can see RAM A here on the left has a post-weaning fecal egg count of a negative 99. So he would be considered to be parasite resistant, and we hope he would pass along genes of resistance to offspring. For the duration of the research project, I'm going to kind of refer to them as low cotyledons, being that they have incredibly low values. Ram B on the right, you can see, has an incredibly positive post-weaning fecal egg count EBV, meaning that he's passing along genes of parasite susceptibility to his potential offspring. And then I also love to kind of throw the values down here at the very bottom for the Katahdin breed, just to show you that there is, in fact, a range of parasite resistant and susceptibility, even within the Katahdins themselves. So while Katahdins are somewhat touted as being parasite resistant, we have to remember that within the breed and what we see in NSIP, there's actually a large range for those post-weaning fecal egg count EBVs. So now let's talk about the research framework. It's really a bunch of numbers, and I know that producers always don't love numbers. So instead of sitting here and having to remind you of these post-weaning fecal egg count thresholds, I like colors. I think colors are a really good way for us to represent this. So for those low cotyledons or cotyledons that are going to be parasite resistant, and have a post-weaning fecal leg count EBV of less than -50, they're going to be represented in green because those are the katahdins that we like. On the opposite end of the spectrum, you're going to see katahdins that have a post-weaning fecal leg count EBV of greater than +50. We're going to consider them to be parasite susceptible or high katahdins, and they will always be depicted in red because those are the ones in which we know will succumb to parasitic infections. And then a little bit later in the talk, we kind of get into some of the middle ground there of some katahdins that fall between a -49 and a +49 in terms of their post-weaning fecal leg count EBV. They're going to be represented in yellow, kind of in between the two, and referred to as intermediates or mid katahdins. So, hopping right into our data, I have death loss for the Southwest Agricultural Research and Extension Center from 2018 to 2023. And I know some of you are probably like, wait a minute, in your introduction slide, you didn't arrive on the scene at WVU until 2021. So, this is actually work predicated by Dr. Andrew Weaver, who had the forethought to start to separate some of this death loss based upon sire post-weaning fecal leg count EBV. And what I want to kind of direct us to is that 2018 death loss. So you can see that for the group, we had death loss percentages start to get into some pretty dangerous territories. Um, in that particular year, death loss was actually diagnosed to be due to a high presence of Clostridium type A, which did not have protectiveness under the CD&T vaccine. So, Clos A is a type of clostridial disease that isn't necessarily covered by the traditional Clostridium perfringens type C and D strains. So we saw some staggering death loss that year. We then implemented an autogenous vaccine, and you can see that death loss for that particular production system returned to somewhat more of a normal value. But what's more interesting in looking at that 2018 time point is that if you were sired by a low post-weaning fecal leg count EBV sire, you had dramatically less death loss when compared to a high post-weaning fecal leg count EBV sired lamb. Now, was that due to parasitism? No, it was due to the high presence of a bacterial species within the environment. So this right here proves the fact of wow, when these sheep are challenged with something outside of a parasitic infection, we still see immune protection. So I arrive on the scene, I start to see some of this, and me being in Dr. Bowdridge's immunology class, I started to parse apart that I think some of this death loss that was occurring early on was also due to colostrum's protective nature there early on in life. So we then ask, what is the value of colostrum? Well, I'm sure all of you producers know that colostrum quality needs to be a complete form of lamb nutrition, right? Being the only form of lamb subsistence, it needs to have adequate proteins, lipids, vitamins, minerals. But I, as an immunologist, see colostrum a little bit differently. I think colostrum quality also encompasses how much passive immunity we can transfer over to our offsprings. That's mainly going to be through these antibodies. IgM is this big, large pentamer that surveils our immune system. IgA is going to be a dimerized antibody. And then finally, the one that we're going to really hone in on in this presentation is going to be IgG. So this guy right here. And I'm sure a lot of producers are like, why IgG? And so I love to explain it as at some point in that ewe's lifetime, she will have experienced a pathogen, right? Her immune cells will go out and they will take a little piece of that pathogen. They'll bring that back to regional lymph, where there in the lymph tissue, we will generate highly specific antibody to whatever little piece of that pathogen we saw. That highly specific antibody that we generate is IgG. So think about this as ready-made protection to things in which you has experienced within her environment that she can now readily pass along to her offspring through colostrum. So let's talk about the original breeding scheme. We did this in very small numbers to begin with, and we wanted to make sure that we were somewhat consistent in some things like age. There's a Katahdin hair index, and there's also lambing criteria that went into making sure that we had equal representation. But the one thing in which we divergently selected these ewes for is that post-weaning fecal egg count EBV. So you can see on average, the high ewes had a post-weaning fecal egg count EBV of greater than a positive 101, and the low ewes had a post-weaning fecal egg count EBV of less than a negative 68. So really, really divergent ends of the spectrum there. And then we went and we sampled those ewes at unique time points. One being an early time point, which would be colostrum. That's going to be kind of that first milk. But then we also looked at just regular milk, approximately 30 days post parturition, and then another 60 days post parturition. And so once we averaged out kind of those 18 and 20 ewes respectively, we get something that looks like this. And this was very dramatic. So at every single time point in which we collected a sample, whether it was colostrum or whether it was milk, we saw highly significant differences in IgG, with low ewes or parasite resistant ewes having significantly more IgG within their colostrum and within their milk. But what I want to really hone in on is looking at that early time point, what we call colostrum, or that first milk in which you would produce. Look at how far apart those bars are separated, or those points are separated. We see about a 2.5-fold difference in IgG depending upon if you are a low or a parasite resistant post-weaning fecal egg count EBVU. So we got really, really excited by this. And we're like, oh my gosh, this is great. But there's also one piece of the story that I haven't really got to share. And so I always love to just pop up the pictures because I think a picture is worth 1,000 words. So when I was having to dialyze all this colostrum and get it ready to do all this fancy lab work, I had to touch each one of those samples. And when I was touching each one of those samples, I started to kind of pick up on a trend. And I'm sure a lot of you here over the webinar can see it as well. But within the year one colostrum, there was a pretty stark difference in terms of color. So this orange top tube here is just regular whole cow's milk. I went out and bought it specifically from the store just so that way we would have a good example of color so everyone here can kind of relate this to something that you would see on the regular. And then you can see in the green tube is colostrum samples pulled together from these low or parasite resistant post-weaning fecal egg count EBVUs. And then this middle red top tube is the high post-weaning fecal egg count EBVUs or the parasite susceptible ewes. And if you start to really evaluate this, you're like, that red top tube looks really close to just regular milk. It doesn't really seem like colostrum. It wasn't nearly as dense. It was somewhat viscous and it was really, really pale in color. So once again, as a researcher, I get the bright idea that we need to find a way to measure this because obviously everyone in the webinar can probably see this difference, but as a scientist, it's not just good enough for me to say, well, look at it, see it. We have to put tangible values on that. So the way in which we put tangible values on it is through colorimetry. It's mainly used in kind of food science applications, but we kind of brought it over for some of this just so that way we could run statistics on these chroma or these color values. So you can see L is going to be white to black, A is going to be red to green, which we're not going to talk about. And then that B value is what we're going to consider yellow to blue. And now you can see that we got excited enough at this about this data that we said, okay, we now want to get a colostrum sample on every single U in which we can from the Southwest Agricultural Research and Extension Center. Um, and the unit manager, I thought at first was gonna have a heart attack. When I told him I wanted him to go and collect all this colostrum from every single U out there. But when we started to really analyze the data, he got so excited about it that he was willing to put in the effort. And so I cannot shout out the staff of the Southwest Agricultural Research and Extension Center in Virginia Tech enough. So now, moving forward for this year two, we're going to measure IgG, we're going to measure color, and we're going to have a range of PFEC values. So you can see we've got low katogens, we've got high katogens, and we'll have some right there in the middle. So starting with the color, each one of these dots represents a U. And so the y-axis, or the one that runs up and down, is going to be our yellowness index. So the higher you are in terms of that yellowness index, the more yellow you appear. And then across the x-axis, or the axis which runs horizontal, you can see I've got that used post-weaning fecal egg count EBV tracked across there. And we get this really nice linear relationship. And all the linear relationship means is that as I get more negative in terms of my post-weaning fecal egg count EBV, I get more yellowness within my colostrum. Vice versa, as I get more positive in terms of my post-weaning fecal egg count EBV, we see less yellowness within the colostrum, which is very similar to the samples in which we collected from year one. For those of us who are maybe overwhelmed by all the dots on that graph, we average this out and you can see that low post-weaning fecal egg count EBV dams have significantly more yellowness in their colostrum when compared back to both the mid PFEC and the high PFEC U's. So U's that would be intermediate and susceptible in terms of their parasite resistance. So pretty cool stuff. Doing the exact same thing now, just from an IgG perspective, we once again see that low post-weaning fecal egg count EBVUs have more IgG within their colostrum. But what I really want to point out in this particular slide is look at how consistently the high PFEC, or the U's that are represented in red, have very, very low average IgG within their colostrum. They are very, very consistently low in the fact. We get into the mid PFEC grouping, or those that are kind of in the intermediate or the yellow, and we start to see kind of a smattering. And then finally, you get some low PFECUs that are in fact incredibly high in terms of their average IgG within colostrum. To represent this just as averages, we get a really nice stair step, meaning if you are a low post-weaning fecal egg count EBVU, or you are parasite resistant, you have more average IgG within your colostrum, followed closely by the mid PFEC grouping, or the intermediate group. And then finally, if you are a high PFECU, or you are parasite susceptible, you have significantly less IgG within your colostrum. So bringing this all back together, I showed you in year one, when we took that colostrum sample, I originally represented it as a line graph. This is now just a bar graph, only looking at colostrum. So you see that 2.5-fold difference in IgG between U's which are low in terms of their post-weaning fecal egg count EBV, and U's which are high in terms of their post-weaning fecal egg count EBV. That was on approximately 38 U's. Looking at that across year two, we see incredibly similar results, indicating that regardless of year in which we sampled, we saw very, very similar average IgG values across both parities. So super, super interesting data that it didn't matter. This wasn't a phenomenon of year. This was consistently stamped across year one and year two, in which we collected those colostrum samples, and was consistently stamped across when we collected from 38 U's and when we collected from over 100 U's. So that's kind of how we wrap up the colostrum segment of this talk. But now let's talk about, okay, so we talked about passive immune transfer, but what about active immunity? What about quantifying an immune response? So, specifically down at Virginia Tech at that Southwest Agricultural Research and Extension Center, we have a really nice mating scheme in which they can look at some of this from a divergent mating. So, the only point of this slide is just kind of showing you how we got these values. So, we took low sires, mated them to low ewes, and then got incredibly low offspring or parasite resistant offspring. And then we took high sires, mated them to high ewes, and generated positive offspring or high offspring or offspring that were parasite susceptible. And then they were all managed together at one facility. So once those lambs were born, we looked at blood parameters around vaccination. So lambs were born mid-March to early April. We waited the appropriate amount of time until they were supposed to receive their CD&T vaccine. And you can see they also received a type A booster because we had that really bad death loss from Clostridium type A that one particular year. So they received a CD&T vaccine along with a type A autogenous vaccine at what we would consider to be week 0. And then I went out and collected blood on those ewes weekly for 8 weeks. And then from timing of first vaccine, 4 weeks later, we came back and we did a booster vaccine of another CD&T and their type A. So this is kind of the timeline just to give you guys a sense of what maybe you're doing on your operation. This is kind of how we phrased it for this particular production facility. Now I'm going to pop up the response to that vaccine. So for those of you who don't understand vaccination curves and you're like, oh, what is she talking about right now? I'll go ahead and give you a hint. The low post-weaning fecal egg count EBV lambs, or the lambs that are represented in green, have a beautiful response to vaccination. So you can see on the bottom X timeline, or the one that runs horizontal, upon initial vaccine, we kind of get a little tiny bump in IgG. Nothing too major, but we do see a small response. But then upon booster vaccine, those lambs say, aha, I know what that is, and they generate a ton of amnestic antibody to that particular vaccine, indicating they're better at recognizing that antigen. This is the same concept that goes to you getting your flu vaccine, your COVID vaccine, any sort of two-time vaccine. This is what we want to induce. We want you to recognize that pathogen and generate some amnestic antibody to that. So the low post-weaning fecal egg count lambs do a really good job of that. Now let's maybe talk about the problem, or these lambs that are here in the red. So look at the line from when we initially vaccinated those lambs to that booster vaccine. There's not a lot of change in IgG, right? It takes those lambs a little while for them to figure out what's going on. So because they didn't really respond to the initial vaccine, when we do give them the booster, they can't reach the same level of IgG because they really didn't do a good job of responding to that initial vaccine in the first place. So this data would actually maybe tell us that for those lambs that have less immune competence, maybe we would need to prime them with a third booster to get to the same level of protective IgG as the low counterpart lambs. So just really interesting stuff when it comes to immune competence. How are they going about mounting an immune response? We can essentially prove that they're just not as good as lambs that are parasite resistant. And then finally, I've done an awful lot of talking, and I always love to kind of sometimes just step back. So if I have overwhelmed anyone and you have completely and totally tuned me out, tune back in for this because I think that this is the data in which I get the most feedback from on producers that are just amazed. So this video is worth 1,000 words. I can sit here and show you graphs and I can talk, but this video right here probably lays it out for us the best. So we've gone over passive immunity, we've gone over active immunity, but all of that doesn't mean that your lambs are never going to get sick. And so my next kind of part of my PhD was, well, when those lambs do get sick, can they bounce back better? Do they have less duration and severity of that sickness? And the answer is yes. So to measure that, I took these two groups of Katahdin sheep. I took the low PFEC or the parasite resistant sheep, and I took the high PFEC or the parasite susceptible sheep, and I gave them something that their immune system would respond to. It's called lipopolysaccharide. It's essentially this big red flag, and it makes their immune system go haywire. They get really high fevers, they get runny noses, lethargy, chills, all the things. They act very, very sick. And so we essentially give them this kind of mock respiratory infection, and then I put cameras on them. And from this data, it's very, very interesting when we just talk about how they even act when they're sick. So I'm going to start the videos now and let everyone kind of see what I see. But and originally, just looking at them from the get-go, the low PFEC Katahdins are all standing, right? Everyone's at least standing. You can tell they're maybe not feeling the best in the world, but they're standing. Whereas compared to the high PFEC group, we've got Katahdins that are laid out lateral, and the undergrad Hannah, who is in the pen moving them, you can see all of the low PFEC Katahdins move to her stimuli. Look at the highs. They feel so physiologically ill that they will not even move. They will not act upon their basic prey instincts to get up and walk away. I'm talking these sheep feel so bad that I could bleed them while they were laying on the ground. So, really interesting. I gave them the exact same thing. Why did they behave so differently? And as a producer, when we think about what drives our treatment decisions, some of our treatment decisions are driven by behavior, right? If you've got a sheep that is acting more sick, like the high post-meeting fecal egg count Katahdins, you're more likely to go and administer treatment to that individual, whereas the low Katahdins could be experiencing the exact same pathogen and are just a little less dramatic, so less severity and duration of those sick behaviors. So, wrapping this all up kind of into a nice bow, selection for low post-weaning fecal egg count estimated breeding value in Katahdin sheep through my dissertation would tell us that we will see a reduction in lamb death loss, Katahdin ewes that will produce more IgG and color within their colostrum, sheep that respond better to CD&T vaccination, and also when those sheep eventually do become sick, they're going to have less severity and duration of those sick behaviors. So, from this, we can say that if you have a post-weaning fecal egg count EBV of less than a negative 50, you are more likely to have a high abundance of IgG within your biological fluids. Where if you have a post-weaning fecal egg count EBV of a positive 50, you are less likely to have a high abundance of IgG within your biological fluids. I think the middle ground there is a lot to be unpacked. And these were values that we set all the way back in 2021. So I think there probably is some flex in where those cutoffs lie. And it's going to be really, really important for us to continue to believe in the National Sheep Improvement Program, continue to look at genetic resources in which we can use to make our sheep just simply better. So, with that, I am at the end of my allotted presentation. I will open it up to see if there are any questions. See the chat. I saw a sheep farmer use Brix meter to determine quality of colostrum, like we do with plants. High Brix means that they're high parasite resistance, and sheep farmers say the same with colostrum. What do you think of this? Well, it's funny you should say that. If you go and look up that paper that I was citing, that Bentley 2024 Katahdin colostrum components, I believe it's called, you will see that we did in fact look at bricks in that particular paper, and unfortunately, we could not find a correlation with bricks values and colostrum quality. Does that mean it doesn't exist? Absolutely not. There's plenty of people out there that are using it, but particularly with the Katahdin sheep, and it could have something to do with the color in which we were seeing. We did not see a correlation there with Brix values. Really good question, though.
So I understand lipopolysaccharides and endotoxin, but it seems like that would make a really good proxy test. I mean, like
Yeah.
a challenge test where, in my observation, regularly collecting FEC, like even in Katahdins, is not everyone does it. And, I mean, while the LPS challenge would be not zero risk, it certainly doing that for a day seems a lot lower a bar as far as like producer labor. Have you guys explored that at all?
We not necessarily. So I'll give just from a regulatory standpoint. So while LPS will make sheep very sick, it would also make producers very sick. So if I stuck
Mhm.
myself with LPS, I would have the same reaction as the sheep had. So
Yeah.
it's a good train of thought though, because I think where you're correct in that is the fact of we should be training producers to collect a blood sample because if a producer collects a blood sample around that timing of CD&T vaccine timeline, we can get a sense of how much IgG they generate to that vaccine, and we then can give them, all right, well, you've got some lambs that are responding better to that vaccine than others. But once again, it does require a blood draw, but it is still, I think, better than maybe an LPS challenge. LPS is kind of nasty stuff. I mean, you saw
Mhm.
those sheep they're kind of out of it a little bit for the LPS.
Hmm.
Let's see if I can stop sharing.
Kelsey Bentley, what would you recommend?
Sana, you lost audio there.
Did we lose Sonia?
I think we did.
Uh,
I think she's got a microwave-based, point-to-point internet setup, if I
hmm.
recall. And so Must have had a cloud or something go by. So Kelsey, have you seen other proxies? I mean, I know FEC is the gold standard, right? And I've done plenty of them, but like I say, just other proxies. I mean, I've looked at like seeing if there's any correlational data between like post weaning weight, and there doesn't seem to be. FAMACHA maybe is weak, but it's so subjective. I mean, helping people head towards better PFE scores without actually, and don't get me wrong, like I would love it if it was easy to take fecal egg counts. They're such a torturous process if you do it yourself.
Yeah, I think the answer to your question is we can't change what we don't measure.
Yeah.
So while you're right, I'm certainly, and too, I get a lot of producers that ask, so I run my fecals, what is the value of a fecal egg count that's a zero? Ugh, the answer is we really don't know because did that sheep have a parasitic infection and then get rid of the infection, or were they just never exposed to parasites? So I agree. There's limitations to fecal egg counts in general.
I
I
mean,
think,
I think they work really well if you take a sweep across your flock, like the same contemporary group, and then you can rank them. Now that
yeah,
may not be necessarily the long-term truth, but at least at that moment in time, you can say, hey, this one's 500 and this one's 30,000, that's being a little exaggerating, but you know what I mean, is you can kind of put them in quartiles
yes,
that likely would hold, but it is a relative measurement. I would say
I
I've
agree.
had a ewe die that had an FEC like at the time she was dying, and I'm pretty sure it was of barber pole. It was like 200. It was nothing, but
Yeah.
she had no resilience, and so it just killed her.
It's a very relative measure, and so it goes back to I think the cotton producers in particular have really put a lot of work into the fecal egg count, estimated breeding value, and you see that that has rewarded them in the fact that they do get healthier sheep from that,
Mm-hmm.
and that health extends past parasitic infections. Would they have been able to derive that if they hadn't looked at fecal egg count? I don't think so. So it's challenging, but once again, my boss goes to the fact and kind of our mindset, which once again, we don't have enough labs that are analyzing IgG at the time either. You've got me now that's trained. We can technically do some IgG analysis. You've got Dr. Scott Bowdridge, who is still running some IgG at the moment. Those assays take time and they're hard, and you've got essentially two people in the United States that are trained for how to do them. I think all in all, though, if you're looking for a better measure of health, it is IgG. So getting to a point where we can perfect that assay, make it quicker, make it more obtainable to more producers, and then we can even start to get heritabilities on antibody, and we can drop some of the fecal egg counting.
Yeah, that makes sense.
Dr. Bentley, what would you expect if you had -50 score rams and bred it to the middle or the higher class and results in the offspring as far as how much improvement could you make?
You're gonna get some variation. So you saw that divergent mating scheme, right, in which we were only breeding lows to lows and highs to highs. There's genetic variation in everything. So did that always perfectly work out the way I wanted it to? No. We had some sheep that even though they were bred to be a low, they would maybe fall more towards the middle or vice versa. So I think though the grand scheme of things, let's say I really can't get a ton of genetic information on my U base, but I do know that the RAM himself technically is that -50 or is at least somewhat negative. In theory, we're moving the needle. And so I think that moving it somewhat and having a RAM that we at least know is parasite resistant is really, really vital, especially because we can contribute the RAM to, approximately 60% of genetic influence. So I think all in all, for producers who are maybe like, I can't go out and register, enroll my whole entire U base in NSIP, just even making an investment in your RAM will still move the needle. Is it going to magically make it everything perfect? No. We even had some lambs that didn't necessarily follow the trend, but for the most part, they should. And it has to do with accuracy values as well. Like we had a ram that his accuracy value maybe wasn't as high. We used him anyways. Come to find out, the accuracy was pretty low on him. And so we saw lambs that slipped through and now fell into categories that they weren't supposed to.
Maybe answering that another way, just generally for EBVs, is like just picking the PFEC one. So an animal of 0 is going to be kind of the mean, right? If you're, let's say the sire is -50 and the dam is +50, and this is the genetic component only, it's not your environment. And so you're going to get half from each. So you're going to get a -25 from the sire and a +25 from the dam. So statistically, and again, like she said, the accuracy of the prediction, your offspring should produce 0. That's just the genetic component. If you have extremely high environmental effects, you're going to be one way or another. And that's the whole idea of EBV calculations is as long as you have large enough groups of animals and they're related to other flocks, it's basically it's using statistics to account for your environment. You know, an example would be like weaning weight. If you're feeding corn and I'm feeding thistles, right? As long as we both have a big pile and there's an interrelation between our flocks, the system kind of statistically figures out is like, hey, these are doing way better, but the common links have to be like your offspring and the same sire of my offspring. They can't be different without the environment causing some effect. If that helps answer the question a little bit.
And they're looking at putting some sort of a correction for environment into a lot of NSIP genetic relatedness. So that I think that's Dr. He just retired from Nebraska, but Ron Lewis would be the one that his group is doing that, where we look at the impact of environment specifically on post-weaning fecal egg count EBV, because your environment has a lot to play there. If you don't have a parasitic infection in Wisconsin and you take that ewe and you move her to Georgia, she's far more likely to now her PFEC shifts. I think I saw something in the chat. So RAM genetics with parasite resistance is more important than eugenetics. I didn't realize that. Well, when you contribute the fact of you've more than likely got less RAMs, it's mainly a way to get around. If you don't know your U base, you could at least invest in a RAM that has favorable values, I guess is the term I'll use. So yes, pretty much. If when in doubt, go with the RAM. You know, maybe make an investment in a RAM that we know has some values. And I think that that's a way to get around the whole messiness of people who don't necessarily want to enroll their whole entire flock in an SIP.
Well, and the other thing is NSIP really only serves, what is it, like 8 breeds?
Yeah.
I'm working on my own EBV solvers. Of course, there's the cold start problem, right? And so it would take several years of collection to have actual values that have decent accuracy. But
There is some SNPs though. They've started to do, it's mainly, so we now have genomically enhanced EBVs. So I will say, even if like a few producers began to submit to GEMS and have some genomically enhanced EBVs, that drastically improves your accuracies fairly quickly. But there does have to be a genomic component there, if that makes
yeah,
sense. If
yeah.
you're going strictly off of data, then yes, you're correct. How can we translate this to our flocks? This is interesting info about how to test a RAM for this. Yeah, that's a challenging question. It's a question I get a lot. I'll, sometimes give the talk and they're like, oh, not another NSIP talk. Essentially, if we can select for sheep which are more parasite resistant, we see less negative health outcomes. So that being said, if we've got ewes that we're constantly having to deworm, or we can at least keep a record of which ewes or rams are our problem ewes or rams and get rid of them. We'll often see total herd health outcomes increase. So while, I've given a lot of data on, NSIP and how we use those numbers to run the stats and the statistics, it's important to just know that sheep, which are parasite resistant, are often more healthy. So if you have ewes that are within the same environment and some of them have just look unthrifty, appear to be suffering from parasitic infections, because I know we're not all doing fecal egg counts, but if we maybe suspect, my answer would be to cull hard. And I think that that's a way that we can maybe implement that, just boots on the ground on our own flocks.
Dr. Bentley,
Yes,
could you pull up your line graph that shows IgG versus the parasite resistance again?
I absolutely can. Let me share my slides.
Because if it looks like if a ram truly, say a ram is truly has a high score, there's not a lot of variability. If you go to a ram that has a low score, it doesn't correlate very well to having high IgG.
Was this the graph you were referring to?
Yes, yes.
Yes. So what we are confident in from this graph is yes, if you have a high post-weaning fecal egg count EBV, or you are parasite susceptible, you are almost guaranteed to have a very, very low amount of IgG within your colostrum. Now, look at the left side of the graph, so all of those green dots. Notice the smattering. So we see less consistency within our low PFEC, but there's outliers within there that do have incredibly high values. So just important to note, you're essentially biologically destined for, if you have a low PFEC, you're going to have a low biological IgG within your colostrum if you're a dam. We do see some actual variation though in the low group, which is intriguing because in theory, I would have liked them to cluster a lot higher. But when you look at the average, there's still statistical differences between the three groupings, if that makes sense, because then that gets into this. But it goes back to the whole point of fecal egg count is not necessarily a great measure of that, right? Because we still have ewes that are technically low in terms of their post-weaning fecal egg count EBV. And they also have, it's maybe not the lowest, but I would say an intermediate amount of IgG within their colostrum, if that makes sense. I might have just confused everyone, but it's intriguing. But the only way in which you're going to get that really, really high IgG, so these guys way up here at the tippy top, is through this low PFEC grouping. There's this one little outlier right here, this guy that's throwing things off. But for the most part, I would have liked to have seen them cluster all here. But you can still see there's some ewes that will have some IgG that's relative to this PFEC. But this is the more telling graph. I mean, you start to average this out, and on average, there is a beautiful stair-step effect of IgG across all of these groupings. But there are outliers.
Dr. Bentley, you said that you guys did not observe a high Brix correlation with IgG. I
We
only was
didn't.
asking because I was hunting around and seen a paper, I put it in the chat, that they said they
Yeah.
did see it, which of course that's
Yeah.
science. You see different things.
Yeah,
I just want to make sure I
exactly.
understood earlier correctly.
I tell my grad students that's why it's called research, because if it
Yep,
was,
yep.
we only had to search once, it would just be search. But no, we didn't, and it was disappointing because I expected to give us a shoot side measure of IgG because that's what I wanted to tell my producers. Go get you a refractometer. They're $50. Put a drop of colostrum on this refractometer, and that's going to give you, a relative value for IgG, hopefully. Within the Katahdin breed, at least when we did this across 2 years, we did not see that. So you can see if you're really interested in it, it's that Bentley et al. 2024. It'll be Katahdin colostrum components, I believe, is the title of the paper. It'll have some of these graphs, but then it'll also have some of the Brix refractometry where you can see we did not find a whole lot. And I trust my highly specific assay more than the refractometry, essentially.
Did you? I'm trying to think of my question here. I would say speculatively that the bricks should still correlate with the quality. Like it might still, maybe it doesn't directly correlate with the immune system, like you're saying, but it should still correlate with the milk quality pretty well.
Potentially. We did no nutritional analysis on any of this
Mhm.
colostrum. All of
Yeah.
it, by the time you got the colostrum, to get what we needed in terms of IgG, you essentially had to use the whole sample. So,
Mm.
yes, you are correct. That Brix value could correlate to something like soluble protein, or it could just simply correlate to sugar. So there's a lot of things that are kind of probably solubles in colostrum that are driving differences in Brix, but we just couldn't detect that with IgG.
Right. That makes sense.
Mhm.
Dr. Bentley, in the environmental aspect, did you see any correlation between the animals that perhaps grew faster, maybe consume more, opposed to animals that were medium sized or didn't grow as fast? And I ask that because it seems most of the breeds that are considered parasite resistant seem to be smaller, slower growing animals. Could is it possible that they just don't consume as many um, parasite larvae as some of the other animals, which maybe just eat more and consume more?
Yeah, really good question. So I'm gonna caveat this because once again, there's people out there with data that would directly contradict what I'm about to say. Dr. Dave Nodder, he was my mentor's mentor, would be one of them. And so I'm very cautious, but I've actually presented this data in front of Dr. Nodder and he was intrigued by it as well. Within the lamb crops that were out at the Southwest Agricultural Research and Extension Center, so one particular unique system raising Katahdins in Virginia across multiple years, we have not seen statistical differences in things like post-weaning weight between these divergent groupings. And you have to remember we are making them almost as divergent as possible for their parasite resistance. And we see no statistical difference on growth. Now, let me back that up to say, yes, if you look at breeds that traditionally have more parasite resistance, i.e., the St. Croix, they are not known for their growth, right? They are known for their parasite resistance. Whereas if you look at breeds that are more traditional wool breeds, they are not known for their parasite resistance, but they are often known for their terminal characteristics and growth. So I think this is all a balancing act. So single trait selecting will never work. So if we single trait select for nothing but parasite resistance, you are likely to get maybe some dinky lambs that mount so much of an immune response that they don't grow as well. So when we are actively selecting for these parasite resistant lambs, other things are going into some of these considerations, right? We're not just using post-weaning as the only thing, but we have set up that divergent mating multiple times. I'm going to go to this slide because I want to also point out something. If you kind of look at these sheep in general, the high post-weaning fecal egg count EBV Katahdins have significantly more wool, right? They don't shed out as well. Probably because they have more of the Tunis and Suffolk within their genetics as compared to if you look at this other group over here, just phenotypically, they maybe appear to be a little bit slicker. There's one or two in there that maybe have some traditional wool, but looking at them, they appear to be more predominantly white. Whereas the group here on the left does have some speckles, they've got more wool caps, things of that nature. So just some interesting concepts, right? Of when we select for parasite resistance, we maybe get sheep that look a little bit more hair-like in nature, have probably a little bit more relatedness to the St. Croix. Whereas on the opposite end, when we select for sheep to be parasite susceptible, we see a little bit more of the wool breeds peep out. So just a real fancy way. I'm kind of talking in circles, but the data, at least that we derived from this particular mating, would suggest that we did not see those differences in which you are describing. I can promise you there are papers out there that would suggest otherwise. But in this particular instance, we saw no negative impacts on growth, at least between the two lamb crops across multiple iterations.
Mhm.
I'd speculate it's, you have an immune cost for maintaining a strong immune system that can resist it. But at the same time, if you pay that tax, you actually get to keep all the grass you eat. Whereas if you don't, you end up with all the parasites and they're stealing all your hard work, right? And so you're kind of balancing between the two.
Potentially. It's a resource pie. Everything takes resources. Even antibody is not made of thin air, it's made of protein. So that's why we also see if you increase your supplemental protein, you often decrease your parasite loads. It's most likely because you can increase the amount of antibody that you can generate if you're given supplemental protein. So yeah, everything's interconnected, interrelated. But probably, right? Especially in Southeast Virginia, where it's really hot, humid, and the parasites love to grow. It wouldn't shock me if those lambs who maybe have the genetic potential to be a little bit faster growing are in fact hit a little bit harder with the parasites, and so it knocks them back to what our parasite resistant lambs would be at.
Is there a way to determine an actual difference between the animals that are parasite resistant and the ones that are just more parasite tolerant, where there's no physical signs, but they still may be uh, heavy carriers?
It's hard. It Yeah, the whole resilience is a very interesting concept. I don't have the answers for resilience. It's like a if you were potentially challenged and you could clear that infection, that would be great. And it's maybe the fact of we need to establish artificial infection protocols. And if we gave maybe like a subclinical dose of parasites to sheep that were maybe not previously exposed, it would give us a way to maybe start to parse apart, is it true resistance? Is it just resilience of sheep that can eventually start to combat some of that infection? I don't know. I think at the end of the day, we genetically had a mating scheme that allowed it for us to do really great research. Um, were they resilient or were they simply resistant? Heck if
Yeah,
I
and
know.
that's probably worth highlighting is that those are technically two different traits, resistance versus resilience.
Yeah. So we technically were measuring something that was genetic resistance. Resilience would be if we all gave them the exact same parasitic burden and then started to do fecal egg count reduction or see who could clear that parasitic infection. Technically, they're within the same environment, but it's just a kind of a hard question to answer. I think it goes back to you have to establish artificial infections then to really start to parse apart some of that. And who wants to artificially infect their sheep with a highly pathogenic parasite? No one.
So in that
Questions?
picture,
This has been a very interesting meeting.
Thanks. Janet, did you have a question?
so in the screenshot that you have up there, would that be more an example of resilience than resistance?
No, because we still have post-weaning fecal egg counts on, like we have EBVs on these sheep. So they genetically are either more resistant or susceptible to that infection.
So resilience is you have lots of parasites, but they're not hurting you that bad. You know, like you be able to, I make up some numbers here. Let's say you have 1,000 eggs per gram of barber pole, but you still have a good FAMACHA score, right? Versus another sheep that has the same 1,000 eggs per gram of parasite load, but they're like anemic on the verge of death, right? So one of them is much more resilient to the infection than the other. And resistance is like you gave them both 1,000. The resistant one is going to drive them out, and so you go measure it again a few weeks later. Maybe the high resistance animal, maybe it's like 400, and the low resistance animal, maybe it's 1,200. It went up.
Yeah, it's hard concepts. I think all in all, when we have a genetic correlation, I'm quick to call it resistance. It's either parasite resistance or susceptibility because I genetically, even from like I said, these are genomically enhanced EBVs. We have single nucleotide polymorphism shifts that we know will trend to make that individual either succumb to parasitic infections or be able to resist parasitic infections. So I yeah, throughout the talk, I refer to it as resistance. If we maybe don't have as much of a genetic component, I think that's when it becomes more of a resilience. And that's flock to flock. Goes back to some of those ewes in which maybe had that infection, got rid of it. We would want to keep her around, right? Because she's no longer contributing to our problem. The one that is, we would want to cull her out.
For those that can't see the chat, Dr. Kelsey, I put your Facebook page on there, the K-State Small Ruminant Research and Extension. So do you want to talk about maybe some of the things that you guys post on there?
Yes. So we will share fecal egg counting schools. I've got two coming up on Valentine's Day. So I shared some of that. We've even got club lamb sales. We do have club lambs here at K-State. So just kind of tells people odds and ends of where I'm at. I will do webinars very similar to this. I know the American Lamb Board is looking to post a sheep IQ. I did some lamb death loss talk for that, being that a lot of my PhD work kind of revolved around that lamb death loss sector. So, yeah, if you ever are wondering where I'm at, what I'm up to, the Facebook page is a really good way to keep up with us, or just normal resources. American Lamb Board is a really good one. The American Consortium for Small Ruminant Parasite Control, another great resource that I always love to point producers to, especially when it comes to research-backed knowledge on parasites.
Any other questions? We really do enjoy this. Well, we hope we can have you back to keep track of what you're doing, because it seems like what you're involved in is something that helps all the producers. And if we were talking about this 30 years ago, we'd be talking about predators and parasites. But now it's parasites and then predators. So you're kind of at the forefront here. So I thank you for our group, our visitors, and hope we can hear from you again.
Yeah, thank you all for having me out. I appreciate it so much. Hope you all enjoy the rest of your evening.